Okay, welcome everybody. Thank you for sticking it out for so many days. This is module 7.1, and we're going to be talking primarily about using network models in a multicellular context, although I'm going to talk a little bit also about best practice in model publication, and we'll do a couple of short exercises on that as well. We're going to be focusing on connecting network models to cell behaviors, and we'll do a simple delta notch example today. That's our classic demo of that. So we're going to be putting our networks back in a multicellular context. Yesterday, we only looked at them as free-floating models. Today, we're going to learn how to load them into cells and work with them in cells. Before we get to that, I'm going to show you a tool that I showed in the lunch break yesterday about how to translate antimony back and forth to SBML. It also lets you browse the BioModels database, which could be pretty interesting. I'm going to talk a little bit about model annotation and publication. It may seem that we're far from having publishable biological models, but it's never too soon to think about what is required to write up models appropriately. And then we're going to go back, look at antibody syntax, talk about integrating CompuCell with networks, and we'll do the delta notch example. So the first thing I want to come to is we have talked about SBML a little bit. We haven't looked at it, and we've used antimony. And there is a very convenient web-based tool called MakeSBML that will allow you to convert antimony models to SBML, SBML models to antimony, and also to browse that BioModels database that I mentioned. That will pull up models in SBML, and then you could convert them to antimony. And so I would like everybody to go onto their browser and go to sysbio, GitHub.io, MakeSBML. Okay, so let's do a short exercise on that. The first thing I'd like people to do is go back, open the code they were working with yesterday. We wrote a little sequential chemical reaction. Remember, A goes to B, B goes to C, C goes to D in antimony. I'd like you to cut and paste from Twedit into the left-hand side of the editor here. You'll have to replace the dummy code that's in there now and convert it to SBML and paste it into the chat. So that's not so bad, I hope. Now let's go the other direction. In the little search box here on the top right, you can type keyword terms. Pick a keyword that is of interest to you for a biological reason. If you don't have one, you might try FGF, WNT, CDC42, cell cycle, or circadian would be possibilities. And what will happen when you do that is it may take a while because the communication sometimes with BioModels is a bit slow. And if it really crashes, you may have to reload the web page. Type in there, and it should pull down a list of possible models. Does it do that? Udish says it does. Good. All right, great. So that is SBML. Not very pretty, is it? Now, it may do it automatically, convert it to antimony, or you may have to click this little arrow button, the right to left arrow button, but convert whatever model you loaded to antimony. Now, look at the antimony code. You'll have to scroll through it. Some of that antimony code should be familiar from the things we've done already. Some of it and mainly annotation is going to look perhaps somewhat strange. So Hannah, you're going to have to click the arrow button to convert. So if has anybody tried looking up a model from BioModels yet? I can do it. I can do one as an example. Kyle tried one. Now let's see if we can actually just maybe together look for a second at what you saw. If you look at one of these images, the screenshots that was shared, I'm looking at the one that Nehar shared. You'll see at the top, it says created by libantimony, then it says compartments and species. We haven't talked about compartments in antimony. Antimony allows you to draw boxes around reactions to create what are called compartments. Those are particularly useful when you have to do volume conversions. Then there'll be assignment rules. And I mentioned briefly that you have dynamic assignment with colon equals. You have a bunch of those. Those are basically function definitions, if you like. Although there also is a separate function definition command in antimony. And then you have the chemical reactions that we've seen before, listed below. Eritra's example looks pretty similar. If I look at Eritra's example, I also see species initialization values and compartment initialization values. If you scroll down through the search, you may find some concepts. For example, here, display names, reaction 1 is creation of cyclin. That's an annotation explaining the meaning of what you've done. You also have things like SBO terms, cell.sbo term equals 290. Those are also annotations of identity. You also have units definitions, for example. All of these things are things that in principle should be in a publishable model. Now, Raghu put up a Goldbetter submite segmentation clock. What I see on the screen is mostly things that we have defined already. The equal sign greater than or less than works as an arrow as well as a dash. And so I think it's worth taking some time to explore the different kinds of information that you can see in the SBML. SBML is much more designed for annotation than antimony is, but you can in fact convert back and forth. Some of these features that we're not going to have time to talk about with antimony are documented in that Tellurium Read the Docs, which is quite useful. LLMs are pretty good at understanding those as well, so you can ask. So the last thing I'm going to ask you to do, and this is more challenging, it may or may not work, is take the code that we wrote yesterday in CompuCell, paste in the antimony code that you just generated instead of your simple chemical reaction model. You're going to have to change the plot instruction in that code to plot one or two variables from the antimony and see if it works and give me the results. I'm serious. You probably are going to have to adjust the time scale of the problem. And if it doesn't work, I'm going to give people 3 minutes to do this. If it doesn't work, I'm going to move on and make it a homework problem because there are other things we need to cover. But I think it's a good experience to try to do that. Take the antimony model that you generated using this tool that we've been working with, replace the model in the code from yesterday with that new antimony model. And then when you're doing your plots, last time we were plotting A, B, C, D, or X or Y, you're going to have to change the name of the plot variables, plot one or two variables from the new model, and see what you get. You just got it to work. Congratulations. You got a cell cycle model going, huh? That's pretty neat. This technique doesn't get you off the hook entirely because you have to think about how to interface the antimony model with the things you're doing in CompuCell. But it's a great way to start. So what are the things you have to do? You have to open the CompuCell model from yesterday. You have to copy the antimony from the left-hand panel here. There's even a copy to clipboard button at the bottom. Replace the antimony code in the Tellurium V2 model with the code you've copied. And then look in the antimony, pick the variable, a couple of variable names, and plot them, changing that one line in the CompuCell that will allow you to plot. There we go. It's important that everybody be able to do this, because if you can't do this level of mastery of the work we did yesterday, you're not going to be able to do the exercises going forward. So I wanted to take the time to make sure everyone can do this. I realize this is a bit of a challenge exercise, but I also think it's important to consolidate what we've done so far. Somebody says it's crashing. What's the error message you get when it's crashing? Okay. If you're getting a syntax error, that means that somehow you copied the antimony incorrectly. That's possible. Giovanni got something to work? That looks like a Gillespie version of it, but that's fine. When you paste it in the chat, you might paste the name of the model as well. That makes it easier to understand. Shivani showed to do comparison between the ODE version and the Gillespie version of the model. That's pretty neat. So one thing people may have found, it looks like most people got the timescales to work, is whether the model worked outside of the box. Some of the models in BioModels actually aren't runnable, so it's possible that that can happen. Some of them may have syntax errors in them, although it's a little unlikely for that. The timescale in the antimony load function is definitely something you may have to adjust. I think we'll skip the other one, which I think I'll make as a homework problem for today, which was try loading the original SBML model from the right-hand side. Instead of using the load antimony model, you can use in CompuCell load SBML model. That's in the code snippets. Try that. You could do that in the background while I'm talking if you like. Try that and see if it gives you the same results. If this winds up taking too long, as I said, I think this is going to be a homework problem for you, but it's worth doing that. The SBML model and the antimony model should give you the same results, but we'll have to see if that's the case. All right. The next thing I'm going to talk about is a little bit of principles of modeling. And this will make more sense as we go forward, but it's good to have this in the back of your mind from the beginning when you're doing computational models, which is that science depends on the concepts of scientific reproducibility and extensibility. If somebody publishes a paper, somebody else should be able to read that paper, reproduce the results, and extend them. When you publish an experimental paper, you're expected to publish the experimental protocols in detail, although there is something called the crisis of reproducibility, which means that a lot of biological papers, experimental papers, can't be reproduced effectively. It's true in medical science as well. It might seem that with computer code, it should be easy to reproduce things. Code is code. It's not so simple. A major study in 2021 found that more than half of published computational biology articles couldn't be reproduced. They didn't provide the code at all. The code they provided didn't work. The code they provided did run, but didn't do the things it was supposed to do. There were simulations in the paper that were not described in the publication, and so on. There are a number of acronyms that have come to be used to describe how you should publish things. One of them is FAIR, and another one is CURE. models should be findable, accessible, interoperable, and reusable. That interoperability is pretty hard. Reusability is also pretty hard. They should be credible, understandable, reproducible again, and extensible. Publishing models in that way, it takes effort. CompuCell provides some tools in that direction, but not as many perhaps as it should. Maybe you can help us improve that. When you publish a simulation, there are a lot of things that you should do. And most papers that publish simulations actually don't do all of these things. And we've actually created a little organization called OpenVT, whose logo you see at the top of the slide, which tries to encourage people to do things in this way. And we actually give out an annual prize for the paper that's published that follows these principles most effectively. It's not a huge prize, but it's a few $100s. And you get a prize lecture as well. When you publish a simulation, you should at least include a complete conceptual model. We talked about that the other day, last week. You should also discuss the domain of applicability of the model. No model works over all parameter ranges under all situations. It's very important to try to describe the limitations of the simulation that you're doing and its purpose, why you're doing. On a more technical side, it's really important that you have instruction scripts and parameters to generate every simulation run that was used in your article, and the workflows for generating every figure. A user should literally be able to click a button, download code, run, and generate every figure in your paper. That's not easy to do, but there are papers published that way. I could point to some good examples of that. In addition, because most biological models are stochastic, even if people run the exact simulations as before, you should have archives of all the intermediate data that you generated. And that should then allow the user, the reader, to generate exactly the figures from your paper. And also, when they run the simulations themselves, they can compare statistically whether they got the same results. That's actually pretty important result. That's a lot to think about, and one could spend a whole week discussing how to do this properly. But I want these things to be in the back of your mind as you go forward, because if you're going to use computation going forward, whether it's CompuCell or anything else, these are fundamental principles of good practice in science. And unfortunately, most people today don't follow them. And that means that most modeling papers are not cited effectively and not reused because people don't put in the extra effort to make things reusable. Your papers will have a lot more impact if you make the effort to publish them this way. The second thing I wanted to talk about a little bit was model annotation. We've actually seen in some of the examples that were in the BioModels database code names like mesh D055411. Where did those come from? Terminology, in biology in particular, but more generally in science, is inconsistent and confusing and ambiguous. There are standard reference databases called ontologies that are essentially huge dictionaries that contain definitions for many terms. You will find, when you work with CompuCell, that in the world of spatial biology, spatial relationships in physics, there actually are a lot of terms that don't have standard definitions. So it's not that everything can be defined. If you're really ambitious, you can actually generate new definitions. Most of these ontologies are living documents, and you can, just as with Wikipedia, you could submit requests for improvements. When you create a model or definition, you should annotate that to an ontology definition. So if you have a chemical species that represents FGF8, there should be a tag like mesh D055411 that says what FGF8 means. If you're talking, for example, about immune response or autoimmune disease or arthritis, you should have a tag that annotates that concept. Some of those are easier to do, like species names for molecular species. Some of them are a little bit more vague, like immune response. But to the extent that's possible, you should annotate as much as you can. These tags can be used by LLM systems and other systems to search your models, and also to allow people to pull information from those models for reuse and extension. Finding these models, so these tag names, is unfortunately still not something LLMs are great at. There are standard portals. BioPortal is the standard one in the United States. OLS is the European equivalent, and EBI top level also has an equivalent. And so I'd like people to go to bioportal.bioontology.org, and let's take a look at that. Here on the left, it says search for a class. Type a term that's relevant to the biological problem that you're working on there. Could be anything that you choose. You may find something very useful. You may find something not so useful. What you may very well get is something like this, where I typed in a term, I typed in FGF8, and it says matches in 22 ontologies. So I have 22 competing definitions. It's not so easy to decide which one to use. You have to relatively rapidly develop a bit of intuition about which of these ontologies are useful ones and which aren't. Mesh, Go, Uberon are ones that are pretty useful. There's some others that you'll run into repeatedly. This is still a bit of an art rather than a science. LLMs actually are pretty good at giving you advice on which ontologies to use. When you click on the term, it will open a page with the ontology definition. It's pretty small here, but you can see that. And the term tag here at the top, which is what you'll use for your annotation. Okay, so why don't you try that? Put something in the chat for me from that. I picked one of the BioModels from Kolodenko here, and within that, there are annotations like MKK biological system, and then it'll say HTTP identifiers uniprot. That's a major ontology, P09560. You'll also notice that in the SBML, there is an abstract that describes the purpose of the simulation. CompuCell doesn't have that in a formal sense with a tag pair. We probably should support those tag pairs. I think if you put those tag pairs in our XML, it won't do anything bad. I think you can use them, but I haven't tried. There's a question, what's the difference between mesh, snowbed, and so on? So people who build ontologies, and there are surprisingly large numbers of them, come from different backgrounds and build them for different purposes. And so there are general ontologies like mesh, which is a list of medical terminology. Ontologies in principle can have a lot more structure than just term definitions. They can have a lot of relational information as well. There may be specialized ontologies. For example, people who do radiation oncology may have special uses of terms that they want to call out and to make sure that things that are related to radiation oncology usage are all defined. And so you'll find that there are general ontologies. Mesh would be one, SNOMED would be one. And there are also ontologies that are highly specialized, for example, for toxicology, for oncology, and so on. In general, you want to go for the more general ones, unless you're working in a very specialized area. Another very important ontology is Go, the gene ontology. which happens to be full of lots and lots of definitions of particular gene names, although also biological processes, it's also not very well organized. And so there are some issues with the way Go is built. Go historically was built from the bottom up. People defined terms and then tried to cluster them. And that tends to lead to inconsistencies in the architecture of the ontology. If you're just pulling term definitions, it doesn't matter so much. Anybody pulled an ontology term for me? I can do one in front of you if you like. Well, that didn't work very well. Let's try CDC42 here in the top level. All right. So here under UniProt, that's a dictionary of proteins, or here, cell division cycle 42 is gene with gene product. I'll click on that. It'll give me a definition of it. And it'll give me a tag name ENSG000, lots of zeros, 70831. So that's possible. If I go into BioPortal, and search CDC42, it gives me a gene ontology I've never heard of, biological environmental research ontology. Gives me MeSH, which is a classic one. So let's go, and it also gives me gene ontology. So let's look at the gene ontology definition here. Let's hope it actually loads for me. It does. Okay, and so this has an OBO entry, Go 00711521, and a definition of here, not just the gene, but the complex. So this is CDC42 GTPase complex. And so if you just wanted CDC42 by itself, this would be the wrong definition. This is the complex. Okay. So people can try that out. Again, even if you're never going to use CompuCell, when you're going to publish papers in biology in general, you should be doing this. Every term in a paper you publish biologically should be annotated in this way. And if that seems intimidating, I would suggest just picking the most important concepts. So now we're going to do some antimony coding. Come back to that. And the first thing I want to do is just remind you of antimony usage. And especially remember, if you're using Twedit, that there are the code snippets of the SBML-solver menu, the Python pull-downs. Again, you've got your antimony cheat sheet. I recommend that systems biology textbook of Herbert's. And you've got the read the docs online, all of which are great resources. And our LLMs are pretty good at antimony coding too. Basic syntax. Model is specified inside triple quotes. It's just a string, ASCII string. Chemical reactions are the fundamental statement. Name, optional. Species A, optional, goes to species B, optional. Semicolon, rate, required. Semicolons are the delimiters. Naming a reaction makes J1, for example, available to you. You can write ODEs directly with a accent mark. I strongly recommend not doing that. You do have to remember that in Twedit, everything is under SBML, even though we're not using SBML most of the time. And once models are loaded, they're called SBML, even if they were loaded in another way. So when we want to get SBML integrator global options, that applies to antimony, tellurium, and everything else. And so if you replace the word SBML here by the idea of network, that probably will make more sense to you. I want to remind you about how to integrate antimony with CompuCell. We've done free-floating antimony models. The integration is always going to be through Python. We've already loaded antimony models into the entire simulation as free-floating models. Now we're going to load them into individual cells. All antimony parameters and variables are CC3D dictionary entries. They're generally readable and writable. All of them are readable. Names like J1: are readable but not writable. We're always going to have to time step our antimony models in our step function. And there's a single global time stepper. So if you want to control the time resolution of your submodels, you have to do that when you load the model initially. And you can always choose whether you're going to solve the antimony model deterministically, as a differential equation, or stochastically with Gillespie. Okay, to do this, again, we define an antimony model. Model equals triple quotes, model definition, end quotes. I see that PowerPoint has changed the quotation marks because it does that. We can load multiple antimony models into a single cell, or free floating. We can have different models inside of different cells. They don't all have to be the same. If you are going to have different models, you want to have a different handle for each model. That's important. And you are going to have to remember that step size for each model. Doesn't take care of that for you. In the class today, we're only going to use a single model, so we don't have to worry about having multiple models running simultaneously. But people have already done it when they did the Gillespie and the ODE versions of the model running simultaneously. It's no different from that. It's useful to define at the beginning of your CompuCell a set of conversion factors. For example, seconds to Monte Carlo steps, 5 times 60 seconds per Monte Carlo step, days to MCS, or seconds to MCS, 60 divided by 60 divided by 24, units days per MCS. We should probably have all of those hardwired in CompuCell at the moment. It's a matter of best practice to do that. Now, when you define your SBML step size, you should use one of these conversions here. This is a best practice formalism that's basically meta to the coding. It's not required format. But if you follow a schema like this, your code will be a lot easier to read and remember. You can put your antimony model definition anywhere. It's a good idea to put it after the conversion factors, which should be at the head of your code, and before everything else outside of the steppables classes. That makes it available everywhere. So here I pasted in my model string in the beginning of my code. If the information is presented in the header section of your Python, it's available within all of the steppables as a read-only information. You can't write to it. But then you don't have to worry about scoping between the classes. Okay. We've seen this semitogenesis simulation a lot. We're not going to do that today, but it's interesting because it has quite a detailed set of molecular reactions within each cell. You'll notice that I'm not following my own advice. Here, these molecular reactions are written as ODEs, not as networks. So this is an example of do as I say, not as I do. This paper was written back in 2012, and one of the reasons that I push some of these principles of publication and description the way I do is because of experiences like our experiences with that paper, where that paper wound up being very difficult to replicate, even by our own group. Okay, how do we load antimony models? We can add antimony models to a single cell, and where it says antimony, we could replace that with SBML. We could replace it with CellML. and so on, if we were loading different language specifications. Model string, if we're loading from a string, the name of the string. Model name, that's going to be the handle by which we're going to refer to that model. If we have multiple models, those have to be unique names. Cell, in this case, the cell identity for that individual, and the step size. We can add antimony models to cell IDs. In this case, cell is a handle for the cell. We can add antimony models to cell IDs. It looks the same, except we have a list of the IDs. These will be integers. So we'll add it to cell number 6, cell number 22, cell number 55, and so on. We can add it to all cells of a given type. We can have cell underscore types and a list of all of the types. We can add a free-floating antimony model, which is what we've done so far, and that has a slightly different syntax. It's add free-floating antimony model in this case. Model name specifies the handle for referring to the model from Python, and we'll see that a lot. Typically, it will just be SBML.model in my examples, but you have to remember that that word model will get changed to whatever the handle you gave the name, you assigned when you did the creation, instantiation. Step size is the conversion between the time units. If we wanted to read the antimony model from a file instead of from a string inside of our simulation, we would say model underscore file equals the full path name. And that'll read from an external file. If we want to use a stochastic Gillespie solver, we use integrator equals Gillespie, and that should not be capitalized. That's a typo. Twedit, code snippets, and the manuals have all of these options, of which there are a lot. Okay. Something that I don't use very often, but is possible, is you can define initial conditions for given parameter values or variable names. And that can be done in the add antimony model. You can have initial conditions equals, and then that's going to be basically a dictionary with all the values. Time stepping is very simple. Self.timestep underscore SBML time steps everything. Make sure you don't put that inside of a loop in your step function, because if you do, you'll be time stepping many times per step. You don't want to do that. You can control the details of the integrator with the option setting, relative accuracy, absolute accuracy, and there are a couple of other things like stiff, soft, so on that you can do. In general, for most of the simulations we'll run, we don't have to play with that. If you have simulations where the values of the variables change very, very rapidly in time, you may have to change that. If you get CVODE errors, not because you have a divide by zero or a mistake in your algorithm, then it's because of that. Reading and writing antimony values is easy. Variable equals cell.sbml, the model name you've assigned, the handle you've assigned, and then quote, and the name of the species of the parameter. That's it. We can put that on the other side of an equal sign for assignment. We don't have to do any gets or sets. If we want to read out the full state, which will be a list of all of the different variables and parameters, result equals self.sbml.modelName dot values, open close parentheses, will give you the whole list. Something that I encourage people to do that I haven't talked about so much this year is there's a very useful Python function called dir. And if you print parenthesis dir parenthesis object name close parenthesis, that will give you the entire structure of that object, including all of the methods that are available to use it. It seems funny when you're writing computer code that you might not know what is possible with an object or how an object is structured, but it winds up that that's something that we need to do, especially if we're working with other people's code. And so including in your code a print dir on an object is a good way to understand how it works. So now we're going to do our delta notch patterning example. Delta notch is a classic juxtacrine signaling mechanism between cells that are touching each other physically. It's quite important in development, in wound healing, in homeostasis, and also in diseases like cancer. We've heard people talking about, for example, leader-follower cell behavior. A lot of times that's regulated by delta notch. Delta notch is a cross-inhibitory mechanism that tends to lead to checkerboards or alternating patterns, where you'll have one cell that has a high level of delta next to a cell that has a high level of notch. You see delta notch patterning here in the intestinal crypt. This is in the eye of a zebrafish, in the hair cells of a Drosophila inner ear. So you see it all over the place. There are a lot of good papers on this, and there have been a lot of very sophisticated computational models of delta notch. And Lorenzo has just published in the past few weeks a nice paper on experiments and computation on delta notch. So if you're interested in this, you can work with him and look at the current state of the art in delta notch. Today we're going to do a very simple example just to get started. How does delta notch work? Delta and notch are both transmembrane proteins. Delta on one cell binds to notch on the neighboring cell. When delta and notch bind together, notch sends a signal to the cell with the notch on it. That notch does two things. It's going to be called NICD, notch intracellular domain, because the notch is actually cleaved on the surface of the cell, and the intracellular part goes inside the cell. Activated notch inhibits specialization of the cell, and it also inhibits the production of delta. The result of this is that cells with high notch tend to reduce the notch in their neighbors and vice versa. You start out with a pattern that everybody's about the same, and you wind up with a cell that's got high delta surrounded by low delta cells. The actual molecular signaling that's going on is fairly complicated. We're going to use a highly reduced model due to Collier from 1996, which simplifies things enormously. It's only going to think about active delta and active notch in the cell. It's not going to worry about all of the signaling and induction processes that are going on. Every cell is going to have a delta level and a notch level. Delta on one cell is going to activate notch on the neighboring cell, and that's it. So how does this work? Delta here, dd by dt, is equal to a constant times 1 divided by the notch level. The more notch I have, the less delta I get. So notch inhibits delta inside my cell. Delta also decays. Notch is not stimulated by delta in my cell. It's stimulated by delta in my neighboring cells. So d bar is the average level of delta in my neighboring cells. The more delta I have on my neighbors, the more notch I produced, and notch decays. And then there are a couple of parameter values here, which are a little bit arbitrary, but are the ones that were used in the original paper. So we have three concepts here: notch inside my cell, delta inside of my cell, and delta on my neighbors. So this is not something that can be resolved looking at a single cell. I have to look at my cell and my environment. This is a good example of why you can't understand what happens biologically looking inside a single cell, even if you have perfect molecular detail. Okay, if I run this simulation, this is a very old simulation that's not very pretty. This is notch. You'll see regions of high notch in red and regions of low notch in blue. And the low notch cells would have high delta. So delta cells would be isolated from each other. And you'll see this pattern has some oscillations in it and eventually settles down to a stable state. Lorenzo found that experimentally, most of the time, you never settle down to a steady state. You always have oscillations. Okay, so let's build a computer simulation of this. What are we going to have to do? We're going to build this simple delta notch equations in antimony. We're going to load them into each cell. But then we also have to replace d bar inside these equations with the average level of delta from our neighbors. Now in Morpheus, that average signal from neighbors actually is a primitive concept. In CompuCell, it isn't. We have to do it by hand. And so what are we going to do? At each time step, we're going to find the neighbors of a cell. We're going to iterate over those neighbors and add up their levels of delta. We may want to weight by the contact area of the neighbors. That can be useful. Then we're going to normalize that sum by the number of neighbors or the total contact area to get delta average for the neighbors. Now, we probably have to do a little bit of a check. If our neighbor is medium, we don't want to do anything to it, so we'll have an if cell to avoid doing that. And if we iterate over our neighbors and our cell was isolated, for example, we don't want to divide by 0 when we get the average, so we have to be careful about that too. Then we're going to copy that value of d average that we've gotten in the Python back into the antimony, so we'll overwrite the value of d prime. And then we'll time step the antimony model. And we'll use tracking fields to display delta and notch values. And one thing here that we have to be careful with is that the tracking fields will plot dictionary entry values very conveniently. However, they won't plot dictionary entry values of SBML.model dictionary. So we'll have to copy from the SBML value in the dictionary to a local dictionary for the cell. We should probably fix that so it's automatic, but it's not there. So we're going to develop this model in simple steps. Again, it's useful thinking about the computations, conceptual model and writing it out before we get too started. And then we're going to build the code step by step. And I've basically given you the conceptual model in pretty good detail earlier. When we build it, I strongly recommend start by building the simplest executable submodel. For example, creating the initial condition, making sure that works. Creating the network model and making sure that that works. And then add features one at a time. Also know how to turn off a new feature if it causes a problem. After each extension, you want to run debug syntax, make sure the simulation does what you expect. So, for example, and this isn't particularly delta notch, you might create your cells first to make sure that they work, then add a chemical field and diffusion with no interaction with the cells, make sure that that works. Then have the cells take up the chemical and make sure that that works and the concentration changes, right? Then add, and this is really a cancer model that I'm talking about here, then add rules for cell growth and division as a function of the chemical concentration. And then maybe add the intracellular network to definition without having it do anything to control the cells. And then couple it to the cell behaviors. So try to build your model systematically in ways that you can find the mistakes, because these full models can be very, very complex. Debugging them is hard if you haven't built it in. Okay, so what are we going to do here? We're going to build a space filled with cells. We can use uniform initializer. We're going to type the model in antimony. We're going to add the antimony to each cell. At each Monte Carlo step, we'll loop over each cell, loop over the cell neighbors, make sure that they aren't medium, calculate the average D from the neighbors, set the new value of D inside the cell, and visualize the pattern. And this says exercise 5, set 2-2. I think it's been renumbered. It should be 7-1-2 in the current version. Okay, so here are your instructions. Create a 42 by 42 by 1 simulation with one type of cell. Use volume, local flex, contact energy. Start with a uniform initializer. Make the cell 7 by 7. In the steppables file, give the cells a volume target of 50, lambda volume of 2. Periodic boundary conditions. Write the antimony string corresponding to the model we just did. Load the antimony model into each cell. Initialize the values of delta and notch in each cell from a uniform random distribution between 0.9 and 1. Calculate the D average for each cell. Create the auxiliary fields to display the values of delta and notch. Is that a clear task? Okay, so why don't we do that? We'll do it in steps. The first step is create, using Wizard, 42 by 42 by 1 simulation domain, periodic boundary conditions, fill it with cells all of the same type, enable volume, local flex, contact energy, NeighborTracker, and in the steppable start function, assign each cell a target volume of 50 and a lambda volume of 2. Okay, I want everybody to do that, and you can give me the simulation output in the chat. There's a question on neighbor order. Neighbor order of 4 is fine. It won't matter for this application. NeighborTracker is in the list of tick boxes when you have in Wizard, the one that has volume, flex, and all of the other ones. It should be enabled by default. There were a couple of questions in the chat that I can come back to. Model string should be text coming before the triple quotes. Model string is the handle to which you're giving the string. So it's the variable name in which the model string is assigned. You can give that any name you like. You could call it model 1, zebrafish model, delta notch model, and so on. Second question, what are the roles of alternating cells in the eye and inner hair cells? It's pretty common that you don't want particular functional cells to be in a big clump. They need to be spread out and surrounded by supporting cells to be biologically functional. There's a question, what are the differences between calling via cell ID and cell types? If I want to load a model only into certain specific cells, then I do it by cell ID. If I want to have a convenient way of loading the same model into all cells or all cells of a given type in one command instead of having to loop, I do it by cell type. Okay, Nathaniel shows a code where the cells are disappearing. So in that case, you probably didn't define the target volumes of the cells. Raghu has a picture that looks pretty nice. Doesn't fill all of the space. That's okay. You can have free surfaces if you like. That depends on the settings for the initial condition. Nathaniel says that he's looping over the cells. Try making cell lambda volume a bit bigger, like 5.0. Let's see if that makes a difference. Okay, I'm going to go forward one step. So if you get stuck, 7.1.1 delta notch has the code. You should have something that looks like this. We used uniform initializer. We assigned the cell volumes. Okay, step 2 is write the antimony model. And the basic syntax is going to be J1 colon, nothing goes to, in this case it would be D, equals new, you can write out the name, times 1 over etc. Remember caret for exponentiation and antimony. So please type the antimony model string here. And for D bar, you could call it D bar or D average. Okay, please do that. And you're going to assign the parameter values A equals 0.01, B equals 100, V equals 1, K equals H equals 2. Here's an example if you're having trouble. J1 arrow N semicolon D average to the K divided by A plus D average to the K minus N. J2 colon nothing goes to D semicolon V times 1 divided by 1 plus B times N to the H minus D. D average 0.4, D 0.05, N 0.05, K2, A equals 0.01, 1, 102. You can pull those from the example here. Okay, now you're going to load the antimony string. Self.add antimony to cell types. Model string equals model name, string name. Model name equals whatever you decide to give it. Cell types, just the cell type name. Step size, you can ignore 1. You can get that in the code snippets. It'll give you self-add antimony model to cell types. Model name here, I called it Delta Notch. I called my cell type cell A. You'll have to use your cell type name. And I used a step size 0.2. Please try that. The next thing that we're going to do is we're going to change the initial values of D and N. We're going to reference those by cell.sbml. Whatever name you gave to your model. Parenthesis D and N if you used delta notch names like that. And the uniform random syntax available is np.random.uniform min and max value. So in your initialized condition, iterate over the cells and randomize the initial values. Give me a thumbs up when you've got those done. I'll show the code to make it easier for people who are a bit behind on this. You can type the code as here. If you get stuck, you can download the model. I'll give people 2 minutes to copy the code here. Every time you make one of these changes, you should run your simulation and make sure it still runs. Check for syntax error. Okay, now the key thing, you're going to create a loop over all cells, create a placeholder for the average, loop over the neighbors of the cells, check if neighbor, so that you rule out medium, read the delta value, accumulate the delta value, keep track of the number of neighbors, normalize by the number of neighbors, and update the value D average. If you got that done in a hurry, keep track of the contact area and weight the contributions to delta average by the contact area of the neighbors. If you don't remember how to iterate over neighbors, remember that in the CompuCell Python code snippets under visit, there is visit neighbors. And I'll jump ahead and show the code here that I came up with. Here, I go over my cell list, set average contact to 0, number of neighbors to 0. I iterate over the neighbors of my cell, get the common surface area. I don't remember that, so I use the code snippet to generate the syntax. If my neighbor isn't medium, I add my contact area, my number of neighbors, and I print the result. If you don't want to do the contact area, you can ignore average contact here. Okay. Here's a simpler version of that. The average is 0, number of neighbors is 0. For neighbor in self.neighborList, if neighbor, add up and get the average. Last thing we're going to do is create the tracking fields. Remember, extra fields automatic tracking. We'll have to copy the value from cell.sbml.dn quote n to cell.dict quote n, because the tracking field will only read from cell.dict. Here's code for that in the init. Self.tracking cell level scalar attribute, field name notch, attribute n, and then in the step function, copy cell.dict d equals cell.sbml dn d. Give people a minute to do that. Again, if you're stuck, please go to the exercise, download it, and please put in the chat your result. Now you've got the complete simulation. Here's what that should look like. Again, you never have to type these long names. You pull them from the Python snippets in Twedit. And that's the full simulation. So I'd like people to run that simulation and put their results in the chat. There's a question. So adding d over n from SBML to dictionary happens in the step. So it's every time step. Yes, you have to do that every time step. If you weren't able to get the code yourself, please download the code and open the code that's there. There's a question about what's causing the patterning. The patterning is caused entirely by juxtacrine signaling. There's no diffusion across membranes. That's correct. Nathaniel says, my delta notch fields are empty. Double check that you copied from the dictionary with the antimony values into the general dictionary for the cell. Again, if you're having trouble, look at the exercise 7.1.1, delta notch shows the result. When I do these exercises, I always try to have an extension exercise like doing the contact area weighting, so that if some people take a little more time, the people who do it faster can keep going on it and explore it some more. Here's what you should have gotten. Looked like that's what people got. I want to talk now about a CompuCell feature that isn't absolutely essential. And so it's okay if the people in the breakout room don't hear this one, but it's a useful thing to have, which is that CompuCell lets you change Python variables from pop-up windows. And we're not going to do the exercises here because we're short on time, but this kind of thing is very useful if you want to build user-distributed code, something that a non-specialist is going to run. The widgets that we have for doing steering are not the most elegant ones. If you do the Jupyter Notebook deployment, which we'll see later in the week, you'll see how to do that in a more elegant way. But for today, this is what we're going to do. And I'm not going to have time to have us do these exercises today, so I'll ask you to download exercise 7_1_2 delta notch steering. To create a steering panel, and this is in the Python Stepables Twedit snippet, you have to do three things. You have to create the steering panel window, define the parameters and variables to be modified. You have to define a function, a callback, that watches for panel updates and reports them, and then you actually have to go in and change the values of the parameters. And this is event-driven coding, and I don't know how many people here have done event-driven coding before. Probably some of you, not all of you. But it's a little bit different from normal procedural coding. To create a steering panel, this is one of these things that goes in that position after the init, like the tracking field. There's a function def add a steering panel, And within the steering panel, you add the parameters that you're going to update. The variable name, the initial value, the minimum value, the maximum value, the number of decimal digits you're going to specify it with, and whether it's going to be a slider or a pull down. There are a lot of other fancier widgets, but the player only supports these. The next thing that you have to do is you have to define a callback process function, which has a specific name, process steering panel data, and it pulls the new values of the variables or parameters that you've changed. These are local inside of this def, and so you're going to have to transfer them to a cell attribute or global to be able to make them accessible outside. You can also put calls to the steering panel in a step function. For example, if you want the simulation to stay paused until a user selects an option or sets initial conditions. So for example, here the step function, you could say, I'm going to hold the simulation until the type is determined, while self.get steering panel parameter simulation type is select simulation to run, pass, that will do nothing. If I select the simulation type, then I decide what type it is that I start the simulation. So I could actually use a pull down in the steering panel to allow a user to start the simulation in one of different modes. And so we're not going to have time today to do these exercises. I'd encourage you to do them as homework assignments. Here we have the delta notch reaction here, and I want to be able to change the values of A and B, the coupling strengths, in the simulation on the fly in the antimony. And we're going to update the values of the parameters in the antimony model based on what happens in the steering panel. Now, in principle, it would be nice to be able to do that on a cell-by-cell basis, but we're just going to do that for all of the cells at once. So what we're going to do is in the initialization, we're going to add a steering panel that's going to read the values of A and B. Their starter values are 0.01 and 100. That's what we had as our default. We're going to allow the values to be between 0, in this case 0.02 and 200. Decimal precision for B is 0 because it's effectively an integer. Decimal precision for A has to be 3 digits because 0.01, we need 0.001 resolution. The next thing we're going to do is define our callback, our processing function. A and B are read from the steering panel. Then we're going to go through all the cells in our cell list and update the values of A and B. And if people can download and run this, please do that and see what happens when you change the value of A and B. Maybe put the result in the chat. Get something that looks like this. You'll also have a steering panel pop-up that lets you select the values of A and B. I'm sorry we don't have time to do the coding example as an exercise, but I do encourage you to look at the code on your own. Give people just a minute or two to try that out. Steering isn't something that you need necessarily if you're going to write simulations for yourself, but if you're going to use a simulation in a classroom setting or you want to have a non-specialist run the simulation, it's essential that they not have to change the code. And so this is a way for them to dynamically change values with a user interaction. That really was the end of the lecture. You'll see that if you change the values of A and B substantially, and this is something you really have to do at the beginning of the simulation. Once the pattern is established, it doesn't make much difference. But at the very beginning of the simulation, if you change the values of A and B, You could have situations where you don't get patterning or you get everything all delta or all notch, depending on how you set it. This is really not so much because it's a biologically interesting question, but more because it shows you how to use this tool. It's pretty helpful for user interface. Okay, we're not going to have time to do the rest of these exercises. In reality, Notch acts as a promoter of cell growth. If you go back to your simulation, and this is now a homework problem, and start with just 2 cells, and have the cells grow slowly to constant rate and then divide at constant doubling volume, how does the pattern evolve? And you'll see that cells on the surface of the cluster behave differently from cells in the center. Now, if you have the cells divide and the growth rate depend on the level of notch. Suppose you have very low value of notch, the cell doesn't grow, high value of notch, the cell grows. Then you can get rather complex patterning. And I ask people as a homework exercise to go in and try that one out. You'll get something that looks more like this. And again, you have those available for download. It's also interesting if the cells are not in a blob, but are in a chain, for example. As I mentioned in tip selection, for example, for endothelial cell migration, the cells at the beginning and the cells signal to each other with delta notch signaling. A cell that's in a cluster has more delta notch signaling than a cell at the tip, and therefore cells with less delta notch can become leader cells. The cells with more delta notch become follower cells. And so you can explore different arrangements of cells and see which ones tend to get high delta and which ones tend to get high notch. And that actually is quite important in a lot of developmental and healing processes. There is a pretty model of calcium oscillations. I know somebody asked about calcium oscillations earlier in the class that we've done based on work by a group at Notre Dame. In this case, we actually do have propagation of calcium between cells, and the calcium leads to contractile behavior of the cells. Fundamentally, the model is almost identical to the one that we've done here, except that the intracellular model is a little bit more complicated, and the signal transduction between the cells is a little bit more complicated. We're not going to have time to do that today, but in the 2023 version of the course, lecture 8.4 goes over all of that and has the demos. If you're interested in calcium signaling and the coupling of molecular levels to cell motility or cell contraction, I encourage you to take a look at that example. So that's all I had for today. I'm going to put up the poll. I know we covered a lot of material. You do have all of the code available to review, and I'd encourage you to do that.